Hi, my name is Michael Jones, and I'm going to talk a little bit about Ulota crispa. Ulota crispa is a bryophyte, specifically a moss. Now, being a moss, it has no vascular tissue, that being xylem and phloem. It also has a sporic life cycle, which causes it to alternate generations. During this alternation of generations, the gametophyte is the conspicuous phase, and it is haploid. Ulota crispa specifically is very resistant to desiccation. A little bit about bryophytes. They're the most primitive plants in kingdom Plantae. They include liverworts, mosses, and hornworts. This phylogeny over on the right demonstrates showing that the liverworts are more primitive than the mosses, followed by the hornworts. Bryophytes evolved from protists and had to adapt to life on land. Some of these adaptations include an apical meristem, multicellular embryos, spores, an epidermis, and alternation of generations. So focusing on the mosses specifically, we see that they're in the phylum Bryophyta. They have no true leaves, no true stem, or no true roots. They do, however, have rhizoids to help anchor to different surfaces. Now, what kind of surfaces can they grow on? Trees, like Ulota crispa does, rocks, roofs, buildings, even concrete sidewalks. The lack of roots and vascular tissue allow this. Now, over here we have a picture of a moss growing on a tree. And if you can guess it, yep, it's Ulota crispa. Now, I mentioned earlier that the life cycle of Ulota crispa is sporic, and it alternates generations. So, what is a sporic life cycle? Basically, it means that the product of meiosis is spores. And alternating generations means it goes between a haploid gametophyte phase and the diploid sporophyte phase. So over here is a little diagram of the life cycle. We start with a zygote, which is diploid, forms the sporophyte, which is also diploid, and then we go through the process of meiosis, and we know that the life cycle is sporic, so the product has to be spores. And those are haploid. Now from the spores, we form the gametophyte. And the gametophyte is haploid. It has male or female gametophytes. Now when the male produces the sperm and the female produces the egg, they come together through fertilization, and a zygote is formed, and the cycle repeats. So here's a nice picture of Ulota crispa. It's actually a zoomed in version of the one we saw earlier of it growing on the tree trunk. If we zoom in even farther, we can see the gametophyte, which is all of the green tissue that we see, and the sporophyte, which are these brown structures coming out from the green tissue. Now, both of these structures are green and photosynthetic until the sporophyte reaches maturity, and then it turns that brownish color. And they're even photosynthesizing through the winter. So crispa comes from the Latin word for curled. And in this zoomed in image, we can see that the leaves are all curled up. And this happens when the moss is dry. So dry air, the leaves curl up and they condense in on each other, and this allows for high tolerance to desiccation. Now, whenever water is available, like through rain or if the humidity goes up, those leaves will uncurl and they look a lot different, as we can see on this image on the right. So, some literature about the desiccation tolerance and recovery of Ulota crispa was studied by Brown and Buck in 1978. They focused on intracellular potassium levels. They showed that as the plants were rehydrated, they lost a lot of potassium. And specifically, they showed that Ulota crispa is extremely tolerant of desiccation because as it rehydrated, it lost less potassium than many other mosses. And they also showed that it has a high recovery index, so it can recover from desiccation faster than others. Proctor in 2003 also studied desiccation, but used more modern techniques. He used something called chlorophyll fluorescence as a measurement, and this confirmed that Ulota crispa has a high desiccation tolerance and recovery. It was not shown to be as profound as Brown and Buck thought. So here's a table from the Brown and Buck results. Now they showed that Ulota crispa had the highest desiccation resistance value at 52% relative humidity. They also showed that at 0% humidity, it was the third most desiccation resistant. Now for the recovery, they showed that it had a very high recovery, although it was not the highest. So here's some graphs from the Proctor paper. They used some of the same species as brown and buck, as we can see in the two graphs to the left of the Ulota crispa graph. In this study, these two species were more desiccation tolerant than Ulota crispa. However, in the brown and buck paper, they were shown to be less desiccation tolerant. So even though the two studies disagree on how high the desiccation tolerance for Ulota crispa is, both show that it was moderately desiccation tolerant. And this is very important for this moss. So here is a video of a rehydration of a dried out Ulota crispa under the dissecting microscope. You can see the leaves uncurling.
There's literally nothing but
It's a very, very different looking plant.
there's literally nothing but
So Mitchell et al. In 2004 studied the nitrogen response of Ulota crispa. What they showed was that Ulota crispa was not as affected by increasing or decreasing levels of nitrogen as other mosses. There were no changes in growth, there were no changes in tissue nitrogen. Other bryophyte species were affected. So this is a great indication that Ulota crispa may be insensitive to atmospheric nitrogen changes. These are very powerful results. The distribution was studied by Wang and Jia in 2012. So the distribution of Ulota crispa was previously restricted to the northern hemisphere. They showed that 3 other Ulota species that were previously described in the southern hemisphere were not morphologically different from Ulota crispa. This means that the distribution is wider than previously thought and now extends into the southern hemisphere. These are some results from the Wang and Jia study in 2012. On the image to the left, we can see that the rows A, D, and H are all the 3 different plants previously thought to be different from Ulota crispa. Now looking at the morphology through the different structures, such as the whole plant, the capsule, or the leaves, we see that they're very, very similar. On the image to the right shows the electron micrographs of specific tissues and also the spores, which don't look all that different from each other. These pictures help to solidify the results, showing that these 3 plants were not morphologically distinct from Ulota crispa. Heegard and Hangelbrook also studied the distribution of Ulota crispa in 1999. They had a little bit different approach, and they showed that Ulota crispa responds very strongly to the actual tree substrate conditions. So in short, this means that the species that it grows on does matter. And specifically, it grows best on species with stable bark, moderately nutrient-rich, and non-acidic, along with older trees having greater diameter. So I'd like to thank Tiffany Cabrera for helping take the photos of Ulota crispa with the dissecting microscope and also shooting the video of the rehydration. I would also like to thank Dr. Skanga for the use of her dissecting microscope, her input and ideas, and final review of this video. Thanks for watching.